Hi there, this is Dr. Laura Jones, and today's video is on methylation, a very complicated subject, but so important and one that I care a lot about. So let's dig in. We're going to actually spend a lot of time discussing all the precious details of methylation, but let's start with a quick and simple explanation of what it is. Methylation is a simple biochemical process involving the transfer of 4 atoms from one substance to another. 1 carbon atom and 3 hydrogen atoms, and this is called a methyl group. The transfer of methyl groups from one substance to another influences how certain molecules act within our bodies. Methyl groups are transferred and donated between so many different molecules, changing their structure and their function, and by doing so, methylation acts as a switch, turning genes on and off. Methylation is important for so many different transactions to happen in the body. It's needed to create DNA and RNA and regulate our gene expression. It's involved in creatine production, which is used to create skeletal muscle contraction. It's involved in mitochondrial function, which of course influences our energy production and our fat metabolism. It's even used in immune system function, our vascular health, and our cell membrane repair in every cell of the body. It's involved in neurotransmitter production and neurotransmitter metabolism. So in this way, methylation affects our mood and our sleep and our cognition. It's necessary for detoxification and metabolism of medications and our hormones. So when optimal methylation is occurring, it has a significant positive impact on many biochemical reactions throughout the body, which regulate various activities throughout our cardiovascular, neurological, reproductive, and detoxification systems. How does methylation happen? Well, methyl groups are made up of 1 carbon, 3 hydrogen, and provided to the body through a universal donor called S-adenomethionine, also known as SAMe. We're going to use SAMe just to keep things simple and the name shorter today. So SAMe readily gives away its methyl group to other substances in the body, which enables all the benefits that we see when the cardiovascular, neurological, reproductive, and detox systems perform their functions through an array of critical enzymes. Unfortunately, the system that produces SAMe is reliant on one switch being turned on by a critical B vitamin known as methyltetrahydrofolate. Methyltetrahydrofolate, also known as active folate or methylfolate or 5-MTHF. We'll use the shorter names again for ease. So simply put, if enough 5-MTHF is present, the folate cycle and methylation cycle work really well and really efficiently. So let's take a look at these cycles. Here is a slide that outlines the methylation cycle. And as we see on the left, there's the folate cycle. The center and right is your methylation cycle, and you can see where they combine in the middle and influence each other. As we look at the methylation cycle, we need to remember the value of active folate or 5-MTHF, which is circled in red at the bottom of the folate cycle. Remember, 5-MTHF is the switch that turns on the methylation cycle. And the cycle begins in our blood vessels with folate, B9, obtained from the diet, when an important enzyme called methylene tetrahydrofolate reductase acts on folate and picks up a methyl group. So methylene tetrahydrofolate reductase, also known as MTHFR, which I am sure sounds familiar to you. When this enzyme acts on folate, folate picks up a methyl group, and we're able to actually push it into the methylation cycle and methylate homocysteine. So homocysteine is circled in purple here. When we recycle homocysteine, we turn it into methionine, which is circled in orange up at the top. This is really important. This reaction is so important. Methionine becomes SAMe, circled in yellow, and this is a second methyl donor, as I said earlier, so it's our universal methyl donor that I just spoke about. SAMe acts as a methyl donor, which then shuttles that methyl group off for multiple chemical reactions in the body. So you see that methyl group off to the right, circled in green. And this includes the production of DNA and RNA and turning genes on and off. So methylation is actually really critical to managing homocysteine levels and the synthesis and repair of genetic material. as well as the epigenetic regulation of gene expression. The donation of SAME's methyl group reduces the molecule, which reforms homocysteine, and the cycle begins again. It's important to note that various B vitamins are cofactors for methylation, so they actually make those enzymes involved in this cycle work. And two of those are B2 and B12. So as I just mentioned, the methylation cycle works when it works as nature intended. It positively impacts so many biochemical reactions in the body that influence ability to detox, our mood, our cognition, our cardiovascular risk. And remember, methylation is our on-off switch, often curbing the expression of many genes that aid in staying healthy. For example, methylation might keep a gene related to tumor formation from turning on, so prevent it from turning on. And in that case, methylation may help to prevent the development of cancer. So let's talk about dysfunction in the methylation cycle. What happens when things don't go as smoothly or efficiently as nature intended? Dysfunction in the methylation cycle can occur in the presence of nutrient deficiencies. It can occur in the presence of various disease states and, of course, genetic influences. And we're going to talk about all three of these. When the cycle is not functioning optimally, homocysteine rises. And we know homocysteine is an inflammatory marker that may increase the risk of thrombosis, endothelial dysfunction, causing errors in vascular smooth muscle proliferation and skeletal muscle metabolism. And the increase in homocysteine absolutely can contribute to heart disease. So poor methylation may negatively impact the body's ability to produce and regulate glutathione, which is one of our strongest antioxidants, right, to protect us from cancers, unhealthy aging, oxidative stress. Poor methylation may negatively impact our ability to produce high energy molecules, to regulate neurotransmitters, to repair our DNA, even to sleep well, as poor methylation does not allow our serotonin to convert to melatonin. Let's use that as a segue to talk a little bit more about how methylation influences our neurotransmitter production. So in addition to being involved with the conversion of serotonin to melatonin to aid in sleep, methylation is critical for the production of many other neurotransmitters. This is because of a key factor that I've outlined here on this slide that is involved in the production of many neurotransmitters, and this is called tetrahydrobiopterin, or BH4. As you can see in the reaction that I have displayed here on the slide, BH4 is made from BH2, or dihydrobiopterin, with the help of methylfolate, or active folate. So without methylfolate, we cannot make tetrahydrobiopterin. So why is this important? BH4 is absolutely necessary for the conversion of many amino acids into active neurotransmitters. We can see in the image I have on this slide, BH4 is important for tryptophan conversion to 5-HTP and serotonin. And then, of course, serotonin gets converted to melatonin. Now, a decreased serotonin level results in increased anxiety. It results in increased depression. It even results in an increase in substance P. So this is involved with our perception of pain and a lowering of our pain threshold. BH4 also helps with dopamine production. So it is involved in the conversion of phenylalanine to dopamine through tyrosine and L-DOPA. And low dopamine correlates with a decrease in motivation, a decrease in focus. We actually see higher incidence of ADD and ADHD in people with low dopamine. We also see more depression and a higher risk of addiction in people who have low dopamine, as their bodies are searching for dopamine receptor activation. BH4 also is important for the conversion of arginine to nitric oxide through citrulline. So remember, nitric oxide counteracts the effects of angiotensin II. And so without methylation and without enough methylation, we don't have enough nitric oxide. And we can see changes in blood flow to various areas of the body, which can present as memory concerns or dementia if we're considering blood flow to the brain. This can also present as hypertension or endothelial cell dysfunction, contributing to an increase in cardiovascular risk. Other notable reactions, one that we just mentioned, homocysteine requires the methyl group to become SAMe. Elevated homocysteine correlated with higher cardiovascular risk. It's atherogenic and it's neurotoxic, so it's absolutely essential for health that we are able to convert it back to SAMe. Histamine also requires a methyl group to be metabolized. So without enough methyl donors or adequate methylation, we have an exaggerated histamine response. So we may see these patients present with chronic dermatitis, more allergies, more hives, more asthma. So let's talk a little bit about the causes of compromised methylation. The process of methylation appears to decline with age. So this may be one of the reasons why chronic disease is more prevalent in our older population. But outside of aging, there are three most common causes of compromised methylation that I want to go through. One is nutrient deficiency, another one are certain disease states, and lastly, and a big one is genetic variation. So I want to start with nutrient deficiency. A growing number of studies looking into the relationship between nutrition and methylation suggests that the process relies on specific vitamins and minerals. We saw that, right? In the methylation cycle, there were cofactors next to several of the enzymes on that slide. In fact, deficiency of any one of the nutrients involved in methylation can result in a bottleneck and suboptimal methylation status. A methylated B complex is a really strong consideration and an easy solution for some patients that have methylation dysfunction. Using the active forms of B6 and B2 and B12 and folate can really support methylation very nicely. Magnesium is also an essential cofactor, especially in DNA metabolism, and vitamin D is known to stabilize the structure of chromosomes and prevents DNA double-strand breaks. Really important. So these are some of the nutrients that we use most often to support methylation. Increasing intake of these nutrients may help maintain healthy DNA methylation. And to maximize the benefit of these supplements, we want to encourage patients to take the active forms. So I've highlighted those here on this slide, the active form of the Bs. So we have our 5-MTHF as our active form of folic acid, our methylcobalamin as our active form of B12, our pyridoxal 5-phosphate as our active form of B6, and riboflavin 5-phosphate as our active form of B2. Let's talk a little bit about different disease states that are correlated with suboptimal or poor methylation. Abnormal or low levels of methylation have been linked to many different disease states, but some important ones that have a strong correlation are certainly Alzheimer's disease, ADHD and ADD, autism, different cancers, especially colon, stomach, cervical, prostate, thyroid, and absolutely breast cancer, cardiovascular disease, cognitive decline, depression, and certainly Parkinson's disease. It's not always clear, however, if the presence of the disease itself negatively impacts the ability to methylate or if suboptimal methylation is involved in the onset of disease. This probably varies between disease states and within each individual. I want to spend a lot of time talking about genetic variants related to the methylation cycle. These genetic variants are also referred to as single nucleotide polymorphisms, or SNPs. A single nucleotide polymorphism is a small variation in DNA that affects our genes and in turn affects our biochemistry. So there are several enzymes involved in the methylation cycle, the folate pathway and even the transsulfuration pathway, the first two of which we will cover in this video. But before we dive any further, it's really important that we take just a few minutes and go back to Genetics 101. So let's revisit some basic genetics. Let's remind ourselves of some basic terms and foundational concepts, including what a gene is, a basic unit of heredity passed from parent to child. Genes are comprised of DNA sequences and are arranged one after another at specific locations along our chromosomes, which are in the nucleus of all of our cells. Each human has an estimated 20,000 to 25,000 genes in our body. Now, another term that's very important to revisit is the term nucleotides. Nucleotides are the building blocks of our DNA and our RNA. They are comprised of three components: a phosphate group, a sugar molecule, and a nitrogenous base. The phosphate group provides the stability to the nucleotide. So think of it as the backbone that allows for linking with other nucleotides, which of course creates our DNA and RNA. The sugar molecule is in the center of the nucleotide, and in DNA, this is called deoxyribose, and in RNA, it is called ribose. And then we have our nitrogenous base, which is the part that varies among the different nucleotides, allowing for our genetic code to be programmed. We have different nucleotides for the DNA than the RNA. Nucleotides in the DNA are comprised of adenine, thymine, cytosine, and guanine. And those that comprise RNA include adenine, uracil, cytosine, and guanine. Alleles are another important term. Alleles are defined as one of two or more alternative forms of a gene that is created by a mutation found at the same place on a chromosome. So alleles create variations of characteristics and traits. We have homozygous and heterozygous as terms that are used to describe allele pairs. Individuals carrying two identical alleles are known as homozygous, while individuals bearing two different alleles are known as heterozygous. The alleles can come in different forms, one of which is a SNP, a single nucleotide polymorphism, which I mentioned earlier. For example, sickle cell anemia arises from an allele of the beta globin gene, which has changed from nucleotide adenosine to nucleotide thymine. There are also several alleles involved in methylation. So similarly, we can have these single nucleotide polymorphisms. One tiny little nucleotide gets switched out or substituted. And we're going to talk about each of the most well-known SNPs in some detail here. One last thing I want to reference is an RSID number, as this is a unique label used by researchers and databases to identify SNPs. So it stands for Reference SNP Cluster ID, and it's a naming convention used for most SNPs. So an important one just to reference. You'll see the name of the SNP, which we also often use letters and numbers for, and then you'll see the RS number as well. So let's talk about the single nucleotide polymorphisms involved in methylation. One of the most well-known and frequently discussed SNPs involved in methylation involves an enzyme called methylene tetrahydrofolate reductase, or MTHFR. We're going to call it MTHFR. You probably have heard about it. So MTHFR is a key regulatory enzyme in folate and homocysteine metabolism. It converts 5,10 methylene tetrahydrofolate to the active form, 5-MTHF, which then can donate a methyl group for homocysteine conversion so that we can convert homocysteine back to methionine and back to SAMe. Therefore, the MTHFR uses 5,10-MTHF for homocysteine metabolism and methionine formation. This is really important. MTRR polymorphisms can result in a reduction of enzyme activity. So that means if we have a polymorphism in MTHFR enzyme, then we have a decreased ability to remethylate homocysteine back to methionine. So there's some consequences of that, right? One of which would be an elevation of homocysteine and an increase in cardiovascular risk. I want to talk about the two different SNPs involved in the MTHFR enzyme. So because 5-MTHF is also an important coenzyme in DNA synthesis, SNP in MTHFR may be clinically significant in considering cancer development as well, right? It's important to know just because we have the SNP does not guarantee the defect. So just because we have an MTHFR SNP does not guarantee defects in methylation. It only guarantees that we have a genetic predisposition or vulnerability. So we know now for sure that gene expression is subject to epigenetic influences such as our nutrition or our alcohol consumption or if we smoke or our stress levels. So just because we have the SNP doesn't mean we're going to express the SNP. That's an important thing to remember. The two most common SNPs that we talk about with the MTHFR enzyme are pretty well known. They are C677T and A1298C. I want to start by talking about the C677T polymorphism. This downregulates the enzymatic activity of MTHFR. And this results in a predisposition to lower active folate, 5-MTHF, and higher homocysteine. So a person who has a heterozygous genotype for C677T has a 30 to 35% less enzyme activity. A homozygous genotype, so two copies of the SNP, has a 70 to 75% less enzyme activity. So that's a pretty significant reduction in the ability to create active folate or methylfolate. And so that results in a pretty significant increase in homocysteine levels. So without a doubt, patients who are homozygous for this SNP have significantly higher homocysteine, and we really need to consider that. They have a higher cardiovascular risk, and providing them with some methylated nutrients can actually help them a whole lot. The second SNP involved with the MTHFR enzyme is called A1298C. This mutation does not significantly affect folate or homocysteine. However, a combined heterozygous genotype for both the C677T and the 1298C mutations results in significant elevation of plasma homocysteine. And so remember, plasma homocysteine being atherogenic and neurotoxic, we absolutely want to address it if a patient has both heterozygous for both mutations. Both C677T and A1298C have been associated with many different diseases. So these SNPs are not just related to cardiovascular disease. We also see a higher prevalence of these SNPs in patients with Down syndrome, patients struggling with infertility, neural tube defects, psoriasis, diabetes, and Parkinson's, as well as many different forms of cancer. The predominant predisposing factor in disease association appears to be the elevated homocysteine levels, and it makes sense then that supplementation with methylated folate and folate-rich foods may reduce the risk in these patients who are both heterozygous and homozygous for the MTHFR SNPs. Another single nucleotide polymorphism involved in methylation is one that affects the methionine synthase enzyme. Methionine synthase is another enzyme involved in the methylation cycle, and it's responsible for homocysteine recycling back to methionine. This enzymatic conversion requires a methyl donor, so 5-MTHF, and is activated actually by B12, which serves as a catalyst for the reaction. So therefore, adequate homocysteine remethylation requires both methylated folate and methylcobalamin, which is our methylated form of B12. But there is another necessary step here in this part of the methylation cycle. Methionine synthase reductase, or MTRR, is responsible for maintaining the reduced B12 levels. And so knowing how important both methylfolate and methylated B12 are for methionine synthase, we also need our methionine synthase reductase to be working well for us. So this creates a very unique enzyme relationship between MTR, MTHFR, and MTRR in order to conduct the essential remethylation of homocysteine. I told you this was a little tricky, right? There's a lot of details here. The takeaway is this set of enzymes, known as methionine synthase and methionine synthase reductase, also require methylated folate and B12 in order for us to recycle our homocysteine in this methylation cycle. Now, the A2756G polymorphism that is most common in the MTR SNP within the literature, it has been suggested that this polymorphism may increase enzyme activity, which would actually enhance homocysteine's conversion to methionine, which would actually lead to lower homocysteine levels. So some SNPs are really great, some SNPs are not so great, right? Because we have a SNP doesn't mean it's bad. We actually have some SNPs that help us in a lot of ways as well. There are a few studies that suggest that this SNP actually can result in elevation of homocysteine depending on how it's presenting, or DNA hypomethylation. We need a little more research here. The discrepancies between these studies may be due to the relationship between MTHFR, MTR, and MTRR, and the complex role of epigenetics, so such as those with various lifestyle factors, right? So how is our nutrition affecting us? How is our sleep or our stress affecting us? So in general, it's accepted that this particular polymorphism upregulates MTR and lowers homocysteine levels. We see when we look at MTR SNPs, there's some clinical associations across various populations that correlate to congenital birth defects, or for example, spinal bifida, cleft lip or cleft palate, or cardiac defects. It's really important also to note that certain populations of people are more likely to have more severe implications from these SNPs. So it's a little controversial. This SNP in particular is a little controversial, but still an important one to know about for sure. As I mentioned earlier, methionine synthase reductase, or MTRR, is a very important enzyme in the recycling and remethylation of homocysteine back to methionine. So methionine synthase reductase maintains methionine synthase activity by reducing oxidized cobalamin, or B12, right, and creating it methyl B12 to be used as a cofactor. Genetic variations in methionine synthase reductase lead to a decreased enzyme activity and create a decreased capacity to recycle oxidized cobalamin and vitamin B. So this decreased enzyme activity can affect our ability to methylate by limiting the amount of active B12 available for homocysteine conversion. So this is the most commonly studied polymorphism for methionine synthase reductase, is called A66G. And this SNP has been shown to result in elevations of homocysteine independent of serum folate, B12, and B6 levels. The MTRR gene variant, just so you're aware, is also associated with various cancers, birth defects, metabolic syndrome, mood disorders, along with the elevated homocysteine. This is another great one to talk about. One more SNP involved in methylation that we can see is for an enzyme called catechol methyltransferase, or COMT. This enzyme is actually necessary to break down catecholamines like epinephrine and norepinephrine. It's involved in estrogen metabolism, and COMT SNPs result in a decreased enzyme activity. So individuals with a COMT polymorphism may have an increased risk of inefficient methylation of catecholamines, so inability to efficiently break them down. So we can see actually more anxiety, right, if epinephrine and norepinephrine hang out longer. We also can see they have an increased risk of poor detoxification and metabolism of the estrogens and of toxins. So we can see an increase in cancer risk, especially estrogen-related cancers, with this SNP being present. The V158M variant is the most common genotype of COMT in most populations. Individuals with homozygous positive genotype actually have a 3 to 4-fold reduction in enzyme activity, resulting in vulnerability to poor methylation activity. COMT polymorphisms correlate with mood disorders such as anxiety, panic disorder, eating disorders, as well as aggressiveness, anger, alcoholism, and severity of bipolar disorder. It's also correlated with fibromyalgia and the incidence of migraine headaches. COMT is critical for phase 2 detox of estrogen. So when we have a decrease in that activity, this is negatively correlated with breast cancer health. We can see that there is an increased risk of breast cancer in women with prolonged estrogen exposure or in women with low folate or high homocysteine. So COMT SNPs have been shown to correlate with higher estrogen levels in patients on estrogen replacement therapy, showing us that they are not metabolizing it as well, and so it can build up in the system. So we need to be aware of that, especially when we're perhaps considering using hormone therapy on females. Understanding a little more about their methylation SNPs could be really valuable in helping to make treatment decisions. The last SNP that I want to discuss is related to an enzyme called betaine homocysteine S methyltransferase. We call that BHMT. This enzyme also plays a very important role in the methylation cycle. This is because it converts homocysteine back into methionine, but it does so by using betaine as a methyl donor. So this reaction goes right up the middle of the methylation cycle. So when there is low folate available for methylation of homocysteine, this is a zinc-dependent enzyme that actually can help us out and provide methyl donors in the case of not enough folate. So by doing so, BHMT is actually kind of a backup pathway for folate deficiency to make sure that SAMe production and methylation capacity stay active. So DMG is the byproduct of BHMT methylation, which is converted primarily to sarcosine. So elevated DMG and sarcosine can be used as indicators of BHMT activity, and high levels of SAMe act to inhibit this enzyme in a negative feedback mechanism, which is brilliant because then it would actually leave the betaine and choline needed to make the betaine to work in other parts of our biochemistry. So pretty incredible. So that's the fourth SNP that I wanted to make sure to go through in a lot of detail with you during this video. So now let's talk a little bit about assessing for methylation defects. As a functional medicine provider, I'm not testing all of my patients by any means, but I'm testing patients where I suspect there is some dysfunction and I want to make sure I better understand how to help them. This might be more severe cases or more severe presentations, or it could be patients I've tried to work with and created some treatment plans already and we're not seeing the success that I would expect. So who to test? The methylation panels that are available now come in many shapes and sizes. Some are better than others, but it's quite an innovative test, and it's designed to offer insight into these critical methylation reactions. And so part of these panels involve looking at methylation metabolites, which can be measured in the plasma, and actually also looking at genetic single nucleotide polymorphisms, which we gather samples to do that through a buccal swab in order to get DNA. Providers may choose to run a methylation panel on many different types of patients. I told you who I am more likely to test it on, either patients where I suspect something's pretty significant is going on, or patients I've already tried to make some progress with and we're not seeing what I would hope for. So these include patients with a personal history of cancer or those who know they're at particularly high risk of different cancers, those with cardiovascular disease, chronic fatigue, cognitive decline, neurological disorders. Also really tricky to crack psychiatric or mood disorders. Even a difficult case of anxiety or depression, we can often unlock some secrets by looking at their methylation status and at their SNPs. And lastly, patients at higher risk for chronic disease. So those with a history of abuse, those with a history with particular toxic exposure, or those, like I mentioned just a moment ago, who are considering hormone therapy. So depending on the panel being employed, the methylation metabolites are often measured in the plasma. And then, of course, the kits are then coming with tubes to bring to a lab to collect serum, as well as a swab to collect DNA from the mucosa inside the mouth. Let's look a little bit further and talk about the components of the methylation panels and how we can use these. So one of the very useful components of a well-designed methylation panel is an assessment of methylation status. As you can see on this slide, there are actually 3, but I'm going to talk about 2 commonly employed indicators of methylation status. So the third one I'm not going to talk about because it involves transsulfuration, which is a subject for a different video just because of the length of this one and how involved this is. But 2 of the 3 components that look at methylation status are something called a SAM-SA ratio and then something called a methylation balance ratio. I'm going to talk about both of these, but these assessments basically compare various analytes in an attempt to identify functional disturbances within the methylation cycle. Let's talk about the first one, the SAM-SA ratio. The SAM-SA ratio is also known as the methylation index in the literature and is actually quite well documented with clinical associations. We reviewed SAMe in detail earlier, but it's important to understand that demethylation of SAMe produces something called S-adenosylhomocysteine, also known as SA, SAH. So SA is an inhibitor of methyltransferases and the precursors of homocysteine. So with methylation being primarily dependent upon SAMe production and supply, and the removal of SA, S-adenylhomocysteine, the SAM-SA ratio has actually been used to indicate the likelihood of hyper or hypomethylation. So a decreased SAM-SA ratio indicates that methylation reactions are compromised and suboptimal. So we see that as SAH or SA rises, we probably are having some compromised methylation. So as a provider, we want to first address SAM-SA abnormalities by ensuring sufficient dietary methyl donors, such as methionine and SAMe, and the nutritional cofactors required for clearance of SA and homocysteine, which include B6, B12, and folate. The SAM-SA ratio is an important one and pretty easy to interpret. The good panels make it really easy with nice diagrams, as I'm showing here on these slides. The methylation balance ratio is another common component of some of the more detailed methylation panels, and this represents an expanded SAM-SA ratio that compares 4 methylated metabolites, including SAMe, to 4 unmethylated metabolites, including SA. And this serves as a way to identify subtle methylation imbalances. The methylation panel also evaluates SNPs. in several enzymes involved along the methylation cycle. And I find this incredibly valuable. So not only SNPs in the methylation cycle, also the folate pathway, and often also the transsulfuration pathway. So we did not discuss transsulfuration in this video. As I said, it's another subject for another day, but certainly an important consideration when we're evaluating patients for SNPs and methylation status, as transsulfuration is a biochemical pathway in our cells that connects methylation with energy production and glutathione synthesis. So very, very important. So shouldn't be overlooked. But like I said, it's a conversation for another video. So back to our methylation SNPs. You can see from this slide that our methylation panels that are assessing methylation also assess some of the SNPs we talked about, including the MTHFR, the MTR, and the MTRR, as well as the COMT and the BHMT SNPs. So this is really great information to have about our patients. I ran this panel on myself because it's a great information to have on ourselves to better understand our biochemistries and our predispositions to chronic disease. So with 2+ signs, of course, next to each SNP, that means that we're homozygous positive for that SNP. 2 negatives, we're homozygous negative, and 1 of each heterozygous genotype. SNP's clinical implication depends on the patient population. As I stated before, just because we have it doesn't mean we're going to express it, but we do see that there certainly is ethnic variance. Genotypic SNPs do not always translate to that phenotypic expression through a clinical presentation, but again, we want to keep our eye out and maybe give the patient a little extra support, perhaps with just some methylated vitamins as a simple first step. It's also important to remember that because of the interconnectedness of methylfolate, folate, and the transsulfuration cycles, biochemical abnormalities might not be due to this one SNP we're looking at. It could be due to another one, right? So because they all do influence each other. And there has been some question, especially amongst patients, just because I have this SNP, should I absolutely take methylfolate or methylated B12? And the answer is no. That, how we're expressing that SNP and how much of a role that expression is playing in our biochemistry changes throughout life. And just because we need more support at one point in life doesn't mean we're always going to need it, and vice versa. So that's something to keep us in mind as a provider. Another component of a well-designed methylation panel is evaluation of biomarkers. Biomarkers are grouped based on their significance to the methylation panel in this particular presentation. This one includes ratios of the biomarkers, methyl group donors, methyl group metabolites, and our transsulfuration metabolites. I didn't circle the transsulfuration ones because we didn't discuss them, but you can see I circled some of the biomarkers we've talked about during this video that are related to methylation. It's nice just to take a look at what their status is, how much or how little do patients have of each. Of course, this can also influence our treatment plans. What I really like is that the presentation of these methylation panels can really pull things together for us and actually provide us with some good graphics to try to share with our patients, because this is such a complicated subject. Sometimes pictures and diagrams make it a lot easier for them to understand. So there's certain companies that do an absolutely excellent job. This particular comprehensive summary displays the methylation cycle and the folate cycle with the enzymes and the different SNPs, as well as the various nutritional cofactors, which are, of course, are essential for it all to work the way nature intended. So these panels allow us to learn with more certainty about the patient's needs for nutritional support, such as amino acids and vitamins and minerals, as well as their genetic predispositions that allow us to better guide them with supplement support to improve their health and their quality of life in the presence of methylation defects. I understand that this was a pretty complicated subject. Thank you for hanging in there with me. I hope that you've gathered some good details to help you better understand how methylation assessment for our patients, not all, but definitely some, can be of value. I've appreciated your time. Have a great night. Thank you.
Welcome to Rupa Health, the best place to order, manage, and track results from over 30 different lab companies in one single place for free. It's going to take you under 2 minutes to sign up, and you can order any functional medicine lab for your client in under 30 seconds. Let me show you how it's done. So here's our beautiful interface. I'm typing in the name of my client, selecting the lab that want to order for them, and hitting send. From there, Rupa and their amazing team handle the rest. They email the client, collect payment, and even offer an interest-free 3-month payment plan. We've also built the world's largest library of information about chronic health conditions, the lab tests that can help you find the root cause, and the evidence-based interventions that you can use to help people heal from them. It's called the Rupa Health Magazine. There, we have in-depth articles about almost any health condition you can imagine. And we give you step-by-step protocols that other clinicians have used to help their clients heal and that are verified by evidence-based sources. You should totally check it out, and it'll transform your practice. And we can't wait to see you. So make sure you sign up today at rupahealth.com.